latest update, this is the link. Or if you don't know where this is, then this is a shorter version of the same link for all the zip files for today's session.
Okay, we're going to have a full room, so find a seat and go ahead and get right into the. Get right on in. You're on? I mean, you might want to wait 1 minute. But, or, I don't know if we're going to have any problems. Okay, good.
Okay, good morning, everyone. I think, yeah, maybe just final. The last technical details. I don't know how to do that. Okay, good morning, everyone. Just before we start, there are some files that we'll be using today. I posted this on the discussion board on the LBRN group page and also on Vive. So please download the files and unzip them so that you have all of the internal files in the folders. I'll be referencing them. So again, you can either copy this link or you can go to the organization page and download these files. It would be very helpful if you already have your account pulled up on the platform so that you can follow along on the practical steps. Okay, so today's session is going to be the introduction to RNA Seq. This is a workshop, one of the first workshops that we'll do. We will try to follow a similar process in later workshops. Basically, this is going to prepare you for the online materials. If you've already started taking the online materials, that is good because you now understand what is happening. But I think this will be a little bit more engaging. And I think as you prepare for the future workshops, just have it in mind that every session will have some files. We'll actually work through some examples. We'll look at the outputs. So please have your computers have your accounts ready, all of that information before the workshop. Again, for those that just came in, all of the files for today's session, including this presentation, is going to be on this link. What you need to do is go to this link, download the zip file, unzip it, and then have all of these files ready on your computer so you know where they are. If you have any other questions, you can't access your account or anything like that, Sahil is here in the back. He can help you. And also for any of you that have any technical challenges with the website, Josh is here in the back. He would be the person to talk to. So as you remember, in the previous session, when we did an introduction to this course, we talked about different types of omics data. And we specifically talked about how processing this data, analysis of this data, is going to be the focus of the sessions. And as you can imagine, different methods for processing the data were created at different labs or different universities with different coding languages. So it's an assembly of different methods. And to appreciate how these methods fit together and how do we utilize them to address biological questions, we've assembled them on the platform. And today, what we will do is we will try to test some of these methods on the platform and prepare for these explanations, the projects, and the step-by-step tutorials on the educational site. So the first section over there on the top is called Transcriptomics, RNA Seq. That's going to be one of the sections for processing the data. Then we will take a look at how to just prepare the data from short reads to a table of expression and how to do differential gene expression. The second thing that we'll look at is a little bit of exploratory analysis if we have enough time. And that includes, so we will talk about some machine learning methods in the context of RNA Seq. How do we do things like BCA, explore our data, visualize our data, and then we'll talk about utilities. And in the utilities, we have a couple of methods for interpretation. I don't know if the quality of this is good enough, but we'll see how do we convert the gene IDs that we get in the gene expression table to something meaningful, something that we can look at from a biological perspective. Now, this is an introduction to this series of lessons that could be taken online. And what do they cover? So just a brief overview. We already completed Introduction to Bioinformatics. And the main point that I hope you can take away from that is how big data is changing our understanding of biology. And then because of this availability of data, how do we utilize data mining, so hypothesis-free exploration of that data, to understand specific biological processes and answer some of our questions about that biology. Transcriptomics 1, 2, and 3 is going to walk you through processing the data from raw reads to a table of expression, hypothesis testing methods like differential gene expression or t-test, students t-test, and then we'll use methods like regression and classification and other machine learning methods to explore the data. And the main challenges are separation of signal versus noise and understanding how do we leverage our existing knowledge about some data set for a new data set. How do we use data patterns to find association and correlation between these patterns in data. After we complete these 3 sessions, we'll talk a little bit about single-cell transcriptomics. Single-cell transcriptomics is going to leverage all of these concepts that are explained in these 3 sections, because really it's an application of the same processing and alignment methods, as well as some of the machine learning methods to understand this another level of complexity that comes with single-cell RNA-seq. So in summary, for the whole session, we will be talking about RNA-seq as a step-by-step process of preparing the data, which is processing, right? How do we structurize the data that comes from the sequencer? Then how do we apply different statistical methods to understand what's in the data and do hypothesis testing? And then how do we interpret the results? So once we have them, how do we do interpretation? And this is done in several steps. So each one could be divided broadly. This is very broad kind of overview of what we will be doing. Pre-processing. So how do we eliminate technical variation that we don't want to include in our analysis? And how do we map the reads, the raw reads that we have on what? Are we mapping on the whole genome? Are we mapping on the known transcripts? So different mapping strategies. And then quantification. What are these numbers that we're looking at? And how do we normalize those numbers so that we can compare apples to apples and not be confused by the variation, for example, in gene length or the total number of reads that we have that might be different between different samples. For the statistical analysis, a lot of people tend to jump to hypothesis testing, and probably in a premature way, right? So that Tuxedo pipeline that goes all the way from mapping to differential gene expression lacks certain understanding and appreciation of data, its variation, and how can we normalize that data before we jump to that step. So we'll talk a little bit about exploratory data analysis. How do we explore those patterns in our raw data? We will talk about hypothesis testing in multiple ways. So hypothesis testing could be done with methods like student's t-test. And what are some considerations about false discovery rate, adjusted p-values, and things like that. But also classification is, in some ways, a way to extrapolate our knowledge or our understanding about the underlying biology and explore that in the sense of data. And primarily, we'll be interested in feature selection. That means when we see a difference, how do we understand how genes or their expression level really shows us a quantification of that difference. And then we will talk a little bit about data mining. So again, data mining from several perspectives. One of them is how do we just explore the patterns in the data and how do we interpret those patterns? But also how do we understand associations between different types of data sets and between different patterns that we identify in the same data set or between different data sets. For interpretation, one way to interpret our data is, of course, visualization. We can just visualize and kind of summarize what we see in the data visually. We'll talk about annotation. So how can we use reference databases to annotate what we see? For example, with gene ontologies and things like that. And we'll also talk about novel discovery. So sometimes we're not just talking about what did I find from known things, but how do I actually find something completely new? And primarily that will be relevant for non-model organisms or very severe modifications of what is known and is available in databases. So the first part is processing of that data, and this is really covered in detail in Transcriptomics 1. Processing of data could be summarized in these several steps. So we do pre-processing, and that just relates to how the data is prepared. What is the raw data in terms of the biological sample before it gets into the sequencer? So we want to address some of those technical preparation steps. Alignment, and which files are we using for alignment? What is the reference genome? What is the Gtf file? So an understanding of all of those things. Quantification, how do we measure? What are some methods of measuring the gene expression levels? And then differential gene expression. Now, before we jump into that, in the online modules, we will be primarily using a data set from publication on patient-derived xenograft models, PDX models. But in this session, I will use a different data set. And so the idea is that the methods And your selection of methods or strategies for analysis is going to be tightly linked with what data set you're using. And you'll see how selection of the genome, of the reference genome, or even different mapping and analysis strategies will change between different data sets. So as you go through these workshops in person or in these sessions, and when you do the online materials, you'll see a lot of similarity, but differences because of the data set. So before we jump into the processing and the analysis, it's important to understand what are we trying to study, because that will help us understand the significance of what we're finding. So what we'll deal with today is a data set from a study related to precision medicine in the context of breast cancer. So what you see here is a mammogram. And this spot right here that is highlighted with the red arrow is a tumor or some suspicious collection of cells. And the first objective from a clinical perspective is, of course, to characterize what exactly is this tumor, where is it coming from? For example, which tumor is it originating from? So to understand that, we can kind of dissect and understand the different tissues and try to pinpoint visually or in other ways the source of that tissue. So one way that we can do that is by looking at receptors, so using antibody staining or immunohistochemistry to check for presence or absence of specific proteins. Right? All of this is going to be relevant in the end to the actual data set, so just to give everybody a conceptual overview. Now, the limitation of that is that you can only look at several types of proteins at the same time, and you are kind of getting there to the presence or absence based on availability of antibodies. So that is a major limitation that could be overcome by looking at gene expression, right? So we know that mRNA is going to be produced into proteins, so these are linked things. And so we can look at something like this, which is a gene panel, and it shows us a little bit more information, right? So it shows us, for example, more genes, and it shows us also high expression levels, medium expression levels, or normal expression levels, and low expression levels. And that's where we get the terms from of overexpression, underexpression, and standard expression, or no change in expression. What you see here is a PAM50 panel. So it contains 50 genes over here on the side. So your rows are going to be genes. And your columns, so to say, are going to be subtypes of cancer. So subtypes of breast cancer, sorry. And so what you'll see typically in your gene expression table is something that looks very similar to this. Your columns are going to be samples, and your rows are going to be genes. How is this information useful? What is the purpose of getting this kind of information about a sample? Well, the first useful information that we have is our ability to prognose, to foresee what category of risk does a patient fall into. So for example, what you see here are Kaplan-Meier curves for PAM50 and Oncotype DX. These are two commercially available panels that are used in clinics to understand survival rate and the rate of recurrence for different groups of patients. So you might want to understand whether a patient has a high risk of recurrence or of death or low risk. And based on that understanding, you can now select different treatment strategies. If it's an aggressive tumor that is growing fast and is metastasizing, your primary goal is to suppress that growth and to stop it from spreading, as opposed to a more mild tumor that you might want to be more aware of the side effects of these very harsh treatments. Now, not so long ago, probably in the 1990s, there was an emergence of precision or targeted cancer therapy. And one good example of that is Herceptin. So Herceptin works in the following way. It is an antibody that essentially blocks a receptor and prevents a specific growth factor from entering the cell with that receptor present, and therefore you can stop this proliferation of cells. Now, this gives us a good understanding, so it's actually linked to our understanding of presence or absence of a specific receptor of a protein, but also our detailed understanding of the process. So that growth factor enters the cell and causes different pathways to be activated, therefore causing proliferation, a biological process that we want to intervene in. So this type of understanding really led to a whole range of precision treatment options, and those are actually linked to how we profile patients. And so profiling of patients and understanding of molecular factors that could help us differentiate between patients actually is a key factor in precision medicine. So therefore, the data set that we're using is somewhat of a representation of this whole problem. It's a publication from 2013 that is called, and you have this publication in the zip file that you just downloaded, called Modeling Precision Treatment of Breast Cancer. What they did is they had a collection of different cell lines derived from patients that were grown, and then two things were done. First of all, they were profiled based on their RNA Seq, Exome-seq, ChIP-seq, so all the different types of omics. And at the same time, the same cell lines were treated with different types of treatments, both available clinically and experimental, and inhibition of growth was measured. So they measured the time from when this drug was stopping proliferation of those cells. And so it gives a good insight into effectiveness of a treatment for a specific molecular profile. So in several of our sessions, we will be using this data set. So some of these files are available in that zip. And what I want to just make sure that everybody has the files unzipped, and you can see some of these folders. So you're not going to see all of them because we're not going to cover everything in today's session. But what you should see is expression quantification under input files. So please make sure that you see that. And specifically, the file that we're looking for is samples.svl right here. So if you have this sample, could you just raise your hand if you have that file, samples.svl? All right, so I think that's 95%. That's pretty good. So what is inside this file? Inside this file, all we did was we downloaded the raw FASTQ files and we uploaded them on the server that we're going to be working on. And so you have SRAs, so sequence read archive files, that are represented by paired FASTQ files. So you see the _1 and _2. SRA is an archive, kind of like a zip archive. So you have to extract the SRA archive, and then you get what's inside, which is raw FASTQ files. Now, these FASTQ files come from cell lines. So just so that you have a sense of exactly what the labels would be. As you can see, each cell line has a name, and that name could be associated with a subtype. So what are these subtypes? You have subtypes that have better prognosis, worse prognosis, and kind of different ways of categorizing these cell lines. Specifically for today's session, we will be looking at only a subset of those cell lines. So we will take all of the normal-like or non-malignant cells and cloud and low. So in total, we will have 11 samples, and you can see visually, these are just two examples, they are very different. Okay, so just to explain what are these file types. The first file type that we'll be dealing with are FASTQ files, and I hope that you can see on the screen here a little bit. What it has inside is a sequence of nucleotides with some additional information. So it has information of quality of that read coming from the instrument. In this case, it was sequenced using an Illumina sequencer. And so some of that quality information is included in every line for each read of that sequencer. We will also refer to FASTA files. And so FASTA files are going to be in the reference genome that we're mapping to. FASTA file is the exact same thing, but it doesn't have any quality files. It's just a complete sequence of the full genome. The third file that we'll be referring to is the Gtf file. Sometimes people use the GFF file, but the Gtf file contains information about what's inside the Gtf. What is the beginning and the end of a gene? What is the exon, exon name, gene name, etc. So how do we know what we map to? It's in the Gtf file and in the FASTA file. What are we mapping? So as you all remember, DNA is transcribed into RNA. RNA is then transformed into mRNA, and then mRNA is finally translated into protein. So what we're focused on is mRNAs. And mRNAs actually are somewhat, they get to their final form in a fairly complicated process. A lot of it related to alternative splicing. So how do the different exons are spliced together, and how do they form what we end up seeing as transcripts? So let's take a look at this process in detail and try to understand what exactly are we expecting to get in these raw FASTQ files. So the main question that we are interested in is, what are we exactly measuring when we end up getting this table of expression? So what we're really trying to look at are genes and isoforms, or what is the real thing that we're measuring as opposed to what we use as a reference. So this is a sequence of a gene, and this is recorded in the Gtf file, as you remember, right? So the Gtf file will have the position, the beginning of the gene, the end of the gene, and all of the exons in between, and the position of the exons and the introns. And then we will have this FASTQ file, which is where the raw reads from our sequencer come. And then we have to align the reads to this sequence that we have in the Gtf file. And we have a number of options. So for example, one isoform, as you can see, is a connection between 3 different exons. And then we have isoform 2. Both of them come from the same gene. And then we have different numbers of reads, and you can see that reads could be of different lengths, and they could align differently to that exon. And then we have 2 different conditions, for example. And now we want to try to understand how does one condition differ from another condition by comparing the number of reads aligned to this gene, right? So that is the objective. And just to give you a reference, what does the Gtf file actually look like? So this is an example of what it looks like. You can see the beginning, chromosome 15, for example. You have an exon. It starts from a certain position on that chromosome. It ends in a certain position on that chromosome. And then you can see that it has a name. And so what is that taxonomy of names? You have a general name for the gene, and then you have a variation. So however many numbers of exons you have in that gene, and that's what you get in the final table. So this is to clarify the data that we will be referring to. Now, how does splicing affect our mapping, and why is it important to remember that what we are interested in are junctions between exons? Because they're really the hard evidence that we have exons spliced together. So in the beginning, we will have reads that map to some region. So you know that they fall within the boundaries of a specific exon. And then you will have some unmapped reads, reads that didn't fall really with a good quality on this exon or another exon. However, within the sequence, you have an evidence that this read contains some portion of this exon, some portion of that exon, and this is hard evidence that they are spliced together. So that's how we start to assemble the isoforms. And when we look at the pipeline, we will see how the steps that we take on the pipeline actually represent this kind of process where we map some reads, then we identify exons, then we look at junctions between exons, make isoforms, and then we quantify. Now, there are some exceptions to remember here, and this relates to the strategies that we rely on when we do our study. So if you are looking at known genes and known transcripts, you're really interested in just reads that fall into one of those known regions. But that's not always going to be the case. So real biology is much more complex than how we document it. And so here are some good examples of introns, for example, being expressed under a certain condition, or only portions of exons being present and portions not being present. So what that really means is that we have a limited understanding for exactly what those regions are and how they work together, especially in disease conditions and in novel organisms. And so if we're interested in those types of changes, we should probably avoid mapping on transcripts and focus on mapping on the full genome, and then looking at what types of variations do we see, what kinds of gene fusion do we see, what kinds of differences in alternative splicing, and how do we evaluate even expression of introns and not just exons. And then the interpretation, of course, becomes much more complex because all of our database-driven interpretation is going to be focused back on transcripts and known events that have been documented. So now let's talk about the role of pre-processing and what are we exactly pre-processing. Before I do that, does anybody have any questions about what I said so far, or is everything clear?
Can you clarify the reads again?
What are the reads? So the sequencer produces short reads. It can only read a certain length of RNA. So RNA is translated into cDNA, and then eventually you read that sequence, and you might have anywhere between 20 nucleotides to maybe 200 or 300 long nucleotides. Now, a gene could be several thousand nucleotides long. So if you have several thousands of nucleotides, and then you have a 200 long read, you want to map that read in some region of that it matches with some quality. So you give a threshold of quality, and then you eventually map it in different regions. And the reads stack up in a certain region, and that's what we call, an expression of that region. It could be an exon or some kind of a region that we can know the beginning, the end, and the height. So that would be the level of expression. So these reads represent what we have in our sample, and what we map to is the reference genome. So that would be the FASTA file of the human genome, for example. That make sense? Okay, so let's take a look at this process of technically kind of what happens. So again, we have some portion of the genome. That portion of the genome encodes several genes, for example, gene A, gene B, and gene C. And then in our actual biological sample, only gene A and gene B are expressed. So they produce a transcript. And now what we're interested to do is we're interested to look at the shattered reads, right? So these portions have been found in these short reads. But in reality, this is a little bit more complex, right? So in the preparation, what happens is we have shattering first. Then we have adapters that are attached to the beginning and the end of that sequence. And then afterwards, we do PCR amplification, right? So we need more material than is typically found, and so we amplify the amount of reads that we have. And as a result, we then read something that somewhat represents the original sample, but not exactly. And so because of that, the whole process of analyzing this data reflects this biological way of preparing that data. So the first step is pre-processing, removing those adapters, and trimming known sequences that come as a result of the library preparation. Then we map or we align those reads to the reference genome. And we have several strategies that we already talked about. And then we quantify, so we need to put a number to it. And so we'll talk a little bit about what are the different strategies that we have for quantification. So because you just joined, I just don't want you to be behind, but there is a file or a number of files that we're working from. So if you could download. So this is posted also on the organization page, but this is a short link. What you'll find there is a zip archive for files that we'll use as input during the session.
I recommend you just download the entire zip. Don't try to view them in Dropbox because some of the files are not viewable. And so you want to just download the file. So just grab that entire zip to your computer and then use them that way.
Got it. All right, so we were talking now about pre-processing. And so actually, as we already know what we need to do, let's go ahead and do that on the platform. So if you can all go to server.t-bio.info, what you should see is the areas of analysis. And we will be looking at this top section. Okay, so if you click through, the first part that will come up is this selection on top, and this is the selection that we want to make. So we want to, first of all, we are using a model reference genome, which is going to be the human genome, the latest assembly, GRCH38. And we will input FASTQ files that are pair-end. And pair-end means that the read is being read from both sides. And so we end up having two files for each sample. And then we will merge them. And as you select that on top, and I will do this all with you just in a second here, what you will want to do is select URL bulk upload right there. And that is where you upload that SVL file. Okay, so let's go ahead and do that together. So if I go to areas of analysis, I will then see this top section. And I will change it here to GRCH38. And I will change this one to pair-end. And then we can upload here using URL bulk upload. And what you saw is samples.svl. So after it uploads all the way, we will press on define groups check. And this is what's inside the SVL. So you can see that these SRA archives are paired together. And you have _1 and 2 in each one. The groups are going to be useful for differential gene expression, right? So if I want to compare a number of samples versus a number of other samples in two groups, I can drag them, I can create two groups and drag them appropriately. Yeah.
Sorry, can you go back to the uploading?
Yeah.
Non-set file type not allowed.
Also what?
Non-set file types.
Yeah,
Never
so
mind. Upload bulk.
yeah, so okay, let me do it from the beginning. So I do pair-end. Here I choose GRCH38, upload files and groups, and I don't do add files, right? So this would be if you actually have the FASTQ files, but we only have references. So references go right here, URL bulk upload. You click on that and you select samples.svl. You choose, after it's uploaded, define groups check. And now we have the files listed. After you have the files listed, you could create a couple of groups and drag them into groups, but we're not going to do that right now. We're not doing differential gene expression. We're just generating a table of expression at this moment.
What start page are you on from the main menu?
I am under RNA Seq. So if I go to areas of analysis, it's this first one. So I go here, upload files and add groups, URL bulk upload, samples.svl, define groups check. I can just do cancel.
Can you help me? I can't get.
And then you can see that this has been completed and I can start creating my pipeline. And obviously the first thing and the only thing that's available is start, and you can read a little bit of an overview. And if you want to read this paper, yeah.
Define groups doesn't work for me. I've uploaded the URLs of the question, but then I cannot really define the groups. It gives back that one, but I don't.
Oh.
Check the,
I think it's yeah, this cancel.
So now we can use the files that were.
Okay, so after the files have been uploaded, you just press on start, right? And so the pop-up shows you kind of what's the process, and you can read this publication to read more about RNA Seq. But now, after we say OK, we see some options available to us. So you can probably remember the colors that we used, right? We have blue for pre-processing, which is right here, pre-processing. And you can click on that, and you can understand what that section does. And then if you remember, we had red, so mapping on genome, and all of the things that we talked about, exon detection, mapping on junction, isoform construction. So all of those things are your options, right? So again, you can, for example, click on mapping on genome or on genes. And then after that, we said that we will do expression levels. So we will quantify, and that was green, right? And so expression levels, there are several methods there. And so the question is, how do we quantify? So we'll talk about that in a second. Okay, so does everybody see what I see? Yep. They're in the back. Do I need to repeat this, or are you good?
Yeah, I think we've done a little can you just start from, it's pair in green, right?
Yeah. Okay, so just quickly over here. So single or pair end. Here, organism is GRCH38. Then you go to upload files and groups. And here you don't do add files. You go to URL bulk upload. Okay, you click on that. You find that SVL file. Choose define groups check. You've got all your files listed right here. We don't need groups, so we do cancel, no groups. And now we are ready to do this. So then I clicked on start, and now I have these options. So the most simple way to process these FASTQ files is just to map everything that we have on transcripts. So I can just go from start to bow tie to T, mapping on transcripts. Okay, and that would be my most simple pipeline, because now I've taken those short reads and I map them on whatever is in my Gtf file. And after I've mapped them, so again, if I made a mistake, I just right-click on this button, and now I go back one step. So I can now read about what does bow tie do. And you will notice that bow tie is involved here in many places. And in this specific case, this is bow tie to T. So T stands for transcripts, G will stand for genome. And I can read about the method right here, and if I want the publication, I can click right here. Save, and after I've aligned those reads to my genome, I need to count, okay, in this specific segment, how many are stacked up together. So to do that, it's actually a non-trivial procedure called RSEM. And what RSEM does is it analyzes the overall statistical distribution of reads and counts in terms of how many fell together, not by chance, right? So we don't just count how many, we actually have to use the overall distribution of reads for the whole transcriptome in this case, and then count accordingly. But here we have a few options. So we have FPKM, expected count, and TPM. And we'll talk a little bit about what these mean. FPKM stands for fragment per kilobase per million. TPM stands for transcripts per million. And raw count is obviously raw count. So we're not going to modify the number in any way. So let's keep it at FPKM. Click on save. You can see that after RSEM, I can actually also quantify my exons and not just transcripts if I was interested in that. But for the simplest pipeline, we don't need that.
Okay.
Let's just click on end, click save, and now give this a name. So you can give it whatever you want. Save, and then run pipeline. So it's important to not just save and leave, run pipeline if you want to run it. Right, so let's just run it. And what you will see is the page will reload. And it's important to notice that right here we'll say started. So 1% or something like that, but it will go from prepared to started. You can also go to My Pipelines, and now you can see all of your pipelines, and you can see the latest one is processing, and it's green. So that's an indicator that it's working. If it's black and it says prepared, that means that it's not running. Did everybody get to this point?
Here's back a little bit.
Go back. All right. How far back do we need to go?
I clicked on end and saved it.
Okay, and then just do run pipeline.
Okay. So now the thing's blinking. The arrows are blinking.
The arrows are blinking. You mean the connection arrow?
Yeah,
Yeah, but did you click on run?
I did.
Okay. Does it say running? Okay, I'll just go back to what the pipeline looks like. It's actually just two methods that we've used so far. So it start both types of T and RSEM expression table end. The very basic RNA Seq pipeline that will just give you a table of expression of known transcripts for your reads. Okay, did everybody else get to this point? All right, so let's talk a little bit about what are some other options. So we did this. So this is going to be your very basic and the fastest pipeline. And here's what a standard Tuxedo pipeline looks like. So it utilizes the same concepts, but in a different way. So you can see that we included some pre-processing steps right here, Trim-O-Matic and PCR clean. Then we went over here and utilized, so from high sat or top hat, which are alternatives of the same algorithm, from here all the way to over here is going to be your Tuxedo pipeline. So high sat maps on genome, identifies exons, identifies junctions, and what it produces at the end is the full alignment file. And that alignment file then is used for combining those exons into isoforms and then adding them into a new updated GTF file. So instead of using the reference GTF file, it creates its own GTF file based on those segments that it found to be exons and junctions of exons present in your reads. Okay, then that updated GTF file is used again in your mapping on the transcriptome. But instead of some reference transcriptome, it's the transcriptome of this specific sample. And then afterwards, of course, you need to still quantify the levels of expression. And so quantification could be of the actual transcripts and of exons. So if you're interested into differences between exon expression, that gives you a little bit more of a detailed view. So this is an expanded pipeline. What other pipeline do we have? Well, if we start from bow tie to G, right here, which is just the mapping on genome, we have an option to also visualize our alignment. So if we actually want to go back and scroll through the whole genome and see where the reads are, we can use bow tie to G and then connect it to visualization. But that is essentially the first step that provides us with a lot of opportunities after we map on the genome. And so you can see that we can also use a novel exon detection just based on segmentation, a different option. And then from there, you can creatively analyze the data yourself. For example, in the following way. So you can identify new exons, create a new adjustment to the GTF, then map on junctions using bow tie I, and then create the construction of isoforms using isolasso, merge the GTF adjusted with the known GTF, and then go from there in the same path to actually align them and quantify them using our sample. A full just a visualization pipeline, if you want to visualize all of your reads, will look like this. So bow tie to G, visualization, end. So this is just to give you an idea of what you can do and the different strategies of mapping that you can utilize. Any questions? Okay, so then let's take a look at what would be the results. So here we ran our simple pipeline. Well, actually, before we deal with the simple pipeline, why don't we create a full pipeline using the pre-processing steps, and we'll just have it so you have it saved. So if we go back to the platform, again, I will start from, you can just click here under Create RNA Seq Pipeline because you're already in this section. You can also use this drop-down menu and go to NGS RNA Seq chip, or you can go to Areas of Analysis and go right here. And each area of analysis will have this little informational tooltip if you want to know what that section does. So if I go back and I create a new pipeline, again, I will have the same settings. So pair end, FASTQ, grch38, upload files and groups, URL bulk upload, samples.spl, define groups check, cancel no groups, and now I can build my pipeline, start. I will select from pre-processing, I will use Trim-O-Matic because that trims the adapters. Then I will clean up the replicates that represent some technical variation because they will be highly conserved. They will be exactly the same stacked together. And then from here, I can utilize, for example, HiSat2. From HiSat2, you can see that I only have a few options. So I will use cuff links, cuff merge, and then bow tie to T, and RSAM expression table. And again, here I can choose FPKM, or I might be interested in transcript per million, and we'll talk a little bit about the difference between those. And here I can just do expanded RNA Seq. Okay, so if you want to play around with this, you can run several of these pipelines and then take a look at the different results. Now, this will take some time, right? So don't expect this to be done in 5 minutes. These are actual FASTQ files, and we have 11 of them. Each one is approximately a gigabyte of size. So probably in a couple of days, you'll see the results. So you'll get an email notification. This is done. You'll click through, and you will be able to see the results. Now, if you want to see the results, a pipeline with results, I will give it to you here in a second. Okay, so this is the pipeline. So notice just so how do you find other, for educational accounts, these are shared pipelines, right? So this is only going to work with the pipelines that you create in this course, and it's only going to work for the pipelines that are with files that are uploaded to the server. But basically, you have a number for each pipeline. So right here, you see at the end 3886673. So all of you can find this pipeline, right? So if I take this link, and I'll put it here on chat for those people that are online. If you want to take a look at it, you can go into your pipeline and just change the number at the end and do 3886673 at the end of that link. So 3886673, and you should see my pipeline because here you will already have the output files. You also have the output files in the zip archive that I gave you at the beginning. But this is what you will see. If you open download files, you will see several things. Expression of genes, FPKM, filtered and not filtered. Expression of isoforms. And then you will also see mapping statistics. So these would be mapping statistics, how many reads were mapped in each sample. And you can see a variation there, and of course that will affect what we are trying to measure. And the main thing that we want to look at is the table of expression. So for example, this would be the gene expression table, and this is what it looks like. So if we download this file, so expression genes FPKM, if I just click on it, it will download. And then what I can do is, for example, open it in Excel. So I just drag it onto Excel. It's a tab delimited file. So that means that the columns are separated by tab. And what you will see is that, let me make this a little bit bigger. Okay, this is what you see inside the table. Okay, so, yeah.
So, and I apologize, catching up with some of the live stream questions. How do you know which GTF file to use since there are so many on NCBI and other places?
So each assembly, for example, we're using the human genome. So each assembly will come with its updated GTF file. But typically, any organism assembly, if it has a GTF, that GTF will have a version. So just like we had GRCH 37 and 38, those are versions. And so we decided to use the 38 version. And what you see on the platform are the most up-to-date versions that we have found. But you can also upload your own GTF. So you can download the GTF, and then you will have a FASTA file for the sequence, the GTF file for the positions in that sequence, and then you can upload both of those and use them as reference.
The other question, or if you make your own GTF file like that, won't you lose the annotations that are associated with the already made ones?
So we don't make a completely new one. We adjust the old one with new things that we find. So that's why it's an adjusted GTF that is then merged together. And so you're actually using information from the known and your own together. Hopefully that answers. Okay, so what are we seeing here in this table? As you remember in that visual representation of the gene signature panel, PAM50, the columns are going to be your samples. The rows are going to be genes. So this is an ensemble ID of a specific gene, EN is G. And what's inside is the level of expression. So if we go over here, now we can talk about what do these numbers actually mean, and what are these measurements of FPKM, TPM, or raw counts. So this would be the sample name, this would be the gene ID, and of course that would be the expression level. But in what numbers? What are we using here? So here are some idea of what these measurements actually represent. So FPKM stands for fragments per kilobase per million of mapped reads. So it actually accounts for two things. One is the total number of reads for that particular sample. And the second thing is the length of gene. So imagine you have a gene that is 2,000 kilobases long and 500 kilobases long. Statistically speaking, more reads are going to fall onto the longer genes just because it's longer, right? So that is one way to overcome that difference between genes. The second consideration is, let's say I have a bucket that is 100% full and a bucket that is 50% full. So I just have different amounts that I'm comparing. So just because this one is 100% full and 50% full, I'm already going to expect that in the differential gene expression, a lot of genes from this bucket are just going to have higher expression, even though that's not really representative of the biology, because the biology should be related to what's overall happening in that specific sample. So that's why we want to adjust and normalize the total number of reads using as our baseline measure. So we align them, and then we measure everything related to that measure of the total of mapped reads. Now, some people would argue that the length of the gene does not matter. And that's why it's becoming more popular maybe today, or some would say, TPM as the standard. And so TPM does not account for the length of the gene. It just accounts for the total number of mapped reads. Okay, but essentially, other than that, it's the same concept. It's a way to normalize your numbers so that they are more comparable between different samples. Just to show you what is, how does that relate to our data set? That information is going to be here under mapping stats table. So if I download that file, again, I can open it in Excel, and it gives me a lot of somewhat useful data, but not exactly maybe. But what you can see here is that the first line, okay, is going to be for this sample. Okay, so I can look at it. Utilizes the alignment mapping. So the SAM file is the alignment mapping. And then this, for example, would be my second sample. So now I want to check whether between these two there is a difference in how many reads were mapped. So you can see that this was 74%, this was 76%, 81, 63, etc. So the overall alignment varies. So if you can now clean up this table and just leave the sample names, you can actually see, okay, so let's say I'll take the first four, not to take up too much of our time. And I can create a little bar plot of the alignment between these different samples.
Is it possible to access the raw reads?
Yeah, you can just open the FASTQ file. That's what you mean, right?
So it's available, so you can download that.
The FASTQ files are available. Yeah, I mean, you can, of course, so this would be, you can see that, for example, there's a difference between these two. And so if I'm just comparing the gene expression between those two samples, I'm already going to be expecting some difference that is not related to the difference in the genes that are being expressed. Okay, so does anybody have any questions about this so far? Yeah.
So I have a question on mapping the reads to the genome or the transcriptome. So in your pipeline, there were several tools listed to do the same job. For example, to map the genome, you have STAR, Biotech, and BWA. And to map the transcript, you have BWT, Biotech2. So how do we know which program to choose to map the reads to the genome or the transcriptome when there are several tools listed to do the same job?
Yeah, so the idea is that you can compare. There are many papers that do, comparisons between how well they perform, and they seem to be performing for different tasks a little bit differently. So a lot of people, for example, utilize STAR when they're interested in gene fusion. BWA and Biotech are different algorithms that do the same thing, and it's been kind of shown that Biotech is more accurate. But still they are widely used for all the same kinds of studies. And so the idea of placing them in the same category would be to give you the option to try different things and come up with your own conclusion of what you want for a specific, they all have, the outputs are going to be a little bit different. The methods that they use are a little bit different. And you can search literature for how they compare in terms of benchmarking for speed, benchmarking for accuracy, right? Those would be the considerations that you would have.
I have a question. Does pairing grades have advantage over singling? Or?
Yeah, they do. So the idea is that the sequencers have a certain rate of error. And so when you do the same thing from different ends, it's like you do it twice. And so you have a higher confidence in the actual sequence of that read. That is the major benefit. And then the analysis changes a little bit in terms of how exactly you combine, because they're not the same read from the same side, they're from two different sides. But it gives you also a better sense of that segment beginning and end. So yeah, there is an advantage.
So that's the follow-up. I had the same question, but under what circumstances do we choose the single end analysis? Like instead of choosing paired end, when do you choose a single end?
It's just related to what's your source data. If your data was prepared as pair end, you can use pair end. But if it was prepared as single end, you can't use pair end.
Okay.
So just based on what data you have. Any other questions? Okay, so next we want to talk about differential gene expression. So now we have a sense of what is in the expression table. How do we compare between two groups of samples? So the idea would be exactly the same process. We first do pre-processing, we then do mapping, we then do quantification, and then afterwards we do differential gene expression, right? Because at the end we are comparing numbers, so we have to get to the point of having numbers, and then we can compare those numbers. And this is where it's going to be useful having TPM and FPKM, because that is a way to normalize those numbers and prepare them for this kind of analysis. Now, what is differential gene expression? What are we actually trying to do with differential gene expression? Does anybody want to answer that?
What was the question?
Why would we want to do differential gene expression?
I mean, in a greater context, or?
Yeah,
Yeah, because we want to see differences in cell, I mean, how the transcriptome responds to a disease condition or in another stimulus, and how that affects cell function and.
right. So we want to compare conditions.
Yeah.
Okay, so in the context of our data set, what are the conditions that we have?
Breast cancer.
So breast cancer is a condition of all of them. Right.
Different types of breast cancer.
Exactly. So we're comparing two different subtypes of breast cancer. And so where do we know which one's what? From this table.
From the top, like the,
So which one is 687?
oh, you had the table, you have the samples listing.
Right.
For that.
Okay, so we need to look up the samples, right? So let's map our samples in the same downloaded file under input files and expression quantification. You have this file called cell lines clinical type that tells us actually what these sample code names stand for, right? So if you look at this ID, you can now know which subtype. So what we can do is we can just, for example, color this with yellow, and let's color this with, let's say, green. Okay, so we want to compare in differential gene expression how normal-like is different from cloud and low. So 720, or let's start from the beginning, 687, 688 are normal-like. 697 is cloud and low. So we can do these as green. As yellow. And then we have 697, 703, 714. So all of these are cloud and low. And then we have 716, 720 is, okay, so this one is going to be also green. And then 720 is non-malignant, 721 is non-malignant. So let's make these yellow. $726 is cloud and low. 34 is non-malignant. So what we're really doing here is we are creating those groups, right? So because in the end, we want to know which file to drag where. So we have to have some kind of a reference. And 36 is also cloud and low. So ideally, if what I would want to do for my differential gene expression is organize them so that they are next to each other, right? So I can move this around. Yeah. Okay. Okay. So the idea of differential gene expression would be to see for this specific gene, right? If I select it like this, could I see a difference, right? So now I've reorganized them, and I can see, so it goes up to 34. So these would be normal-like, and these would be cloud and low. So just looking at this table, any idea which one has a higher expression, cloud and low, or normal-like, just by looking at these numbers?
Yellow ones are the normals, right?
Yellow ones are normal-like, and the green ones, so up until here, right? So These would be, all of these are normal-like, all of these are clouded low. What do you think? Which group is higher expressed than the other?
First of all, non-normal one.
So these are
On
what?
average,
These are what?
like higher.
Higher expressed. So by
On
how
average,
much?
like on.
By how much?
If we take the average, we will say that like we need to find out. Because in some cases it's low, very low, like 636 is very low,
17.
Right.
but there is a very high, like 714 is very high. So we need to look at maybe average
Average?
of, yeah.
So average, you think, is a good way to measure?
The overall, for overall look, it will be.
Okay, so I need to take an average, right? And then I'm saying that, somewhere, so let's do that real quick, just so that we get a sense of what's in here. Right, so now I can prepare an average. So this will be average for normal-like, and this will be average for clouded low, right? And so this would be yellow. And I can just do average right here and select all of these. And then I can do the same for this one and select all of these.
The columns are the versions, right?
Huh?
Columns or versions.
Columns are the individual samples,
Oh,
right?
the samples are coming from different.
Cell lines. So these are different cell lines. Okay, so now I have on average 7.5 and on average 10.33. Is there a difference?
It defines your significance level.
Okay, so how do I measure the difference? What would be the difference between one group and the next? T-test. All right, good. We're getting somewhere. So, okay, so now we have the idea of checking. What does t-test actually do?
It tells us that our means are different. There's a significant difference between the average means in our distributions versus the null hypothesis, which is that they're not significantly different.
Right, so we're really now doing hypothesis testing, right? We're starting with, we assume that there's no difference. And if there is a significant p-value, right, a probability that this could not have happened by chance, we say that there is actually a difference, right? So that would be one measurement. So let's do that.
Do you have to test for the normal distribution of data?
Well, right now, no. But first of all, we can talk about, doing some descriptive statistics on our data. That would be a useful step. But right now, we just want to understand differential gene expression. So this will be more similar to this. So my t-test would be for this versus this. So I get a p-value. What's a good p-value? 0.5. 0.5 or 0.05? 0.05. All right, so is there a difference?
No.
No difference. Okay. So that is one parameter that we use. And what is another parameter? Let's say there was a difference. Let's say it was 0.04. What else? How do I then quantify this difference? So we use something called fold change, right? So fold change would be what is the number that I can assign to this difference. So those are the two things that you need to kind of remember for differential gene expression, is that we're going to be using p-values and fold change to measure the difference. And it's also important to remember difference between what and what, right? So your order, your sequence of what you put in one group and the second group is going to be important, because that will be your fold change. It's one between the other. So what would be the fold change here? For example, would be this is the first group, right? Divided by this group. So 0.72. You can also do minus. You can just subtract one from the other. You can also do a logarithm of fold change. So remember that that number is going to be representative of what you decided to do. So now the problem is, so let's say I can do all of this in Excel. So why do I need to do differential gene expression? Well, one of the problems is that I just have a lot of genes. Let's take a look at how many do I have. I have close to 20,000 genes, right? So I can go 1, 2, and maybe select number 500. But at the end, I have a lot of genes that I have to deal with. And then I have to select those genes that are actually have a good p-value and have a high enough fold change that I can actually say that they are differentially expressed, and I can try and interpret that difference. So let's go back. I was told that we need to wrap up, so let's quickly create a differential gene expression pipeline. So let's again select here parent, and here the reference genome. And let us upload the files. Over here, URL block upload. And now we need to create our groups. So let's create 2 groups. And now we need our reference table. Okay. And I want to drag them in appropriate groups. So I will do, my first group will be non-malignant. So I will take the 720 and so I will have 720, 721, 634. So all of these are normal-like. 634. 687 and 688. So all of these are going to be my normal light, and all of these are going to be cloud and low. And I can do continue. And this is for contrasts between segmentation. If I was to use segmentation and identify new segments, I will use contrast, but here we actually just select all. We'll just do that. Let's do start. And now let's do the most simple one again. So let's skip the pre-processing. Let's just go to bow tie to T, R7 expression table in FPKM. And now I have this option to do differential gene expression. So I can choose between edgeR, DSEQ, and DSEQ2. If I was using the cuff merge, cuff links, etc., I would be able to use cuff diff. So let's, for example, do DSEQ2, and you can read about it right here. And end. What would be the output of these different files? And by the way, you can use edgeR, and then you can compare what's the difference. So we're not going to wait for this to finish, but you do have in your output files, right here under output files, you have differential expression. And here you have the output from DSEQ2 and from edgeR. So let's take a look at DSEQ2. Okay, so here's what we have here. We have log2 of fold change. This is the standard error for log fold change. And then we have p-value and p-adjusted value. The same thing would be there for edgeR, but in a little bit different terminology. So you have log2 fold change, log fold change, then p-value plus FDR is actually the p-adjusted value. So you have all the same information here, right? So now that we have this table, how do we actually select those genes that are differentially expressed from this table? For example, we can use the p-adjusted value first, and we can filter for everything that is less than. Okay, so the problem here is that you have these NAs, right? And so it's going to have a problem with those. So let's convert this to number. And to overcome this, we can do the following. We can actually filter out all the ones that don't have their name. And let's just copy all of this. Okay, so now we can, let me make this a little bit bigger. Okay, so less than or equal to, what's a good p-value? 0.05. Okay, let's see how many I have that are different, that have a statistical significance. That would be my first thing to look at, right? So I have a total of 2,250 genes like that. And the second thing that I want to use is log fold change. So I want to, again, filter out the ones that are the most different, right? So 13 would be the fold change between those two groups. And let's say I will go to a minimum of 1.5. I'm not really interested in anything below that. So let's say 1.5 will give me about 931 genes that have a difference, right? So let's just check. What does that look like? So I'll just mark that with color in here.
In the second method, it's kind of bitwise matching, like in the row sequence for each read. Is it? That's how
For
compares. So in the second one, like differentiating.
Differentiating? Well, you first do all the same stuff and create a table of expression. And then you do what we just did in Excel, right? We compare the means, see whether it's statistically
In the
significant.
second one, still we are using that.
Yeah. Well, not exactly, but yeah, the same principles.
Okay.
So let's just check what this gene, so let's take the first one, the most differentiated, and go back to our expression table and see what's inside. So here, I will find that gene. So it's right here, and I can create my bar plot, and here you can already see that the difference is quite obvious, right? So now I've selected a gene that totally has transcription here, but has 0 or close to 0 here, right? If we select a gene that does not have a good p-value, so if we actually take something like this, right, that does not have a good p-value, and we search for that, it was this one, right? So now I see the difference. The difference in these differentially expressed genes would be that on average, within this group, there is a significance and there is a quantifiable difference. So this was, we'll wrap up for today, and that was it for gene expression and differential gene expression. You will find in the online modules some more examples of things that you can do, including normalization and exploration of your data. And in the next workshop, we will work through the rest of the methods and kind of go all the way to the end, to interpretation, to pathway annotation, to gene ontologies, etc. Any questions?
how do you adjust the p-value? I mean, what does it, how do you do
It
that?
accounts for false discovery rate.
We're gonna talk about LBRs and how we calculate LBRs.
Yeah. Actually, in the online modules, I think there's. Any other questions? Okay. Well,
I have one.
yeah.
So when you said about inspect like the mapping statistics, and then you showed that there was variation there, and some genes that you take into account, right?
How do you take it into account?
Yeah, how do you?
FPKM and TPM.
Yeah, so wait, that was my second question. When you after you normalize, then are you sure you can compare those? I mean, that you take off
No.
this variation?
Because that is just one way to normalize, is look at the total number of reads. What you would want to do really is do summary statistics
Yeah.
and look at your moments, look at your skewness, your distribution, etc., and then do normalization based on those. For example, under utilities, you have something that's called quantile normalization, right?
Yeah,
So
like the
yeah,
arrays,
there is
like
a
if you bring down your-
exactly. You do the means, you line the means, and you actually normalize, adjust for its own noise, right? So there's a certain level of noise in each sample, and then you normalize it between them, and between samples you adjust the means.
So are these in the online tutorials?
Yes.
Okay. Thanks.
Any other questions? All right, thank you, and we'll see you again soon.
This kind of the program, you would be able to access and reuse and
So this is a 6 or 9 months program.
Yeah.
So throughout this whole program, you can do that, and you can access it, and you can also request access afterwards from Nayan or Doug, Ramesh.